so before I start, I would, of course, like to thank the organizers again for giving me the opportunity to present my work here. It's really, really great content. It's really a pleasure. So, yeah, I'm interested in the biological origins of learning, and one of the key questions driving my research is how genes, on the one hand, and environmental factors, on the other hand, interact to shape mathematical ability in our brains. And today I would like to present you some data from an ongoing longitudinal study in which we combine genetic data and brain data of very young children to then longitudinally predict their mathematical ability in school. Yeah, before I start, I would like to provide you with some general background information regarding the genetics of mathematical ability. So, one important thing to note here, of course, is that there is no such thing as a single math gene, right? So the picture is far more complex. There are several genes involved here, and the list is getting longer and longer. That's important to know. And the second thing that is very important, especially for me as a neuroscientist, is that there's actually a huge gap between genes or genetic variants and behavioral variants. Yeah, so it is really inappropriate to think about genes, directly acting on our cognition or on our behavior. Instead, there is evidence that what these genes do is that they actually play a major role for brain development. And these genes I'm showing you here today, they are all well documented in literature to really encode proteins in the brain. And now the question driving my research is, how these gene-brain actions might be used to ultimately predict behavior and explain mathematical ability. Okay, so what did we do in our study? So here we took saliva samples of 101 very young children, 3 to 6 years old, and these children also took part in an MRI scanning session. And as I already said in the beginning, we are still following these children. That's an ongoing longitudinal study. But for a subset of these children, we have already psychometric test data available. Now, what did we actually measure here? I think that's important to explain here, given that we have such an interdisciplinary audience. So saliva samples enabled us to simply quantify DNA variation that occurs just naturally in the population. And what you get out of this is essentially a categorical variable with 3 levels, where level 0 simply reflects these DNA variant that is simply most common in the population, that is most frequently found in the population. And then you have an intermediate category, and then category 2 just refers to the variant that is least, that is very rare in the population, that is not that often found. Okay, that's the general logic behind these genotype measures. Now, at the brain level, we were particularly interested in gray matter development. Why is this the case? Well, it's simply because there's quite a bunch of studies that has already shown before that the genes I was talking about play a role for the development of the gray matter. And the gray matter, simply put, is what is basically nerve cell bodies. Not exclusively, there's also axons, white matter in it, but it largely consists of nerve cell bodies. And today I will focus about the gray matter in the outer parts of our brains, in the brain surface. And the measure we took here was so-called gray matter volume. And to obtain this measure, what's the trick? What do we do here? Well, we divide the brain essentially into these small cubes you can see here, called voxels, and for each voxel of the brain, we get a continuous value ranging from 0 to 1, and 0 meaning that's a low gray matter volume, and 1 meaning that's a high gray matter volume. So we have this categorical genetic variable, we have this continuous brain variable, and then the psychometric, with the help of the psychometric test, we could extract percentile ranks of mathematical performance. And here we simply used this established, standardized test instrument that's available in Germany. And this essentially, from this test you can derive two scales. One arithmetic scale in which children simply have to solve very basic arithmetic problems, for example, addition problems, and a visual spatial quantity processing scale, where children have to solve these classical numerosity comparisons, for example. Okay, so as you can imagine, so there are all these genes that went into the test. So we test for many different genes, and then we also run tests in thousands and thousands of voxels in the brain. So it's very important here to come up with very rigorous multiple testing procedures, right, in order to obtain statistically valid results. And it turned out that only one of all these genes survived these rigorous correction procedures. And this was the gene ROBO1. Some of you might already be familiar with it. And now, I have to say that was a positive association. So, now the question for us neuroscientists emerging here is now, so we do not only want to see, okay, there's a gene-brain association, but we want to link this to particular brain systems. And we were very surprised to find this very, very clear effect in a region that is very well established in the brain imaging literature on mathematical processing, which is the inferior parietal cortex. And I have to mention another issue that is very important here, and that's the issue of power in genetic studies. I think all of you have heard about these discussions. So the background is that the effect sizes of these DNA variations that occur naturally in the population, they are usually considered very small. At least if you link these type of data to behavior, they are really small. But on the other hand, there's also evidence in the literature that the effect sizes of these DNA variations on brain measures is much higher, much larger. And that's what we directly confirmed here in our data with a post-hoc power analysis. So it turned out that despite the fact that we only tested 101 children, which is a small sample for a genetic study, we achieved 90%. This makes us to a certain degree confident that these results really can be replicated in follow-up studies. Yet another thing to mention here maybe is that these effects were were relatively independent of very general biological factors, right? So there are differences in age, gender differences, handedness, total head volume. These things were all controlled for, right? Okay, so now what does this tell us? Why is this interesting, this link between this particular gene and the right inferior parietal cortex? How can we explain this effect? So how does this link to brain, relate to brain development? And to answer this question, we have to go into the animal literature. Because ROBO1 is a very, very well-studied gene in the animal literature. It's known to play a key role for brain development, and in particular for a process called neuronal migration. And let's imagine the following experiment that has been done several times in the animal literature. So imagine we are able to manipulate the genetic makeup of mice in utero. So we're looking now at the developing rat brain in utero, and we have two experimental groups. So one group of mice that simply develops typically, so there's no genetic modification occurring, and then there's another type of mice where you essentially downregulate the function of this particular gene, ROBO1. And that's what we'll look at now. So we zoom into the cortex and see what happens. And if you downregulate LOBO1, what basically happens is that nerve cells lose orientation, right, during very early stages of brain development. Because what LOBO does is it tells nerve cell populations at very early stages of brain development where to go in the cortex and where to settle down. And the rationale behind this now, so the link to humans, is that if you carry one of these infrequent DNA variants, that something similar but much more subtle happens at very early stages of human brain. Okay, that's the general picture behind it. Now we have this gene-brain link. The next step, and the decisive step for us, of course, is to link this to behavior, to the standardized test results. And that's what we did. And importantly, there are, of course, there's a rich set of potential sources of variance here that might ultimately explain the performance of these tests, right? So there are some, so we've already been talking about working memory, IQ, and all these factors. And we have control for these factors. So we have control for IQ, for working memory, as I've already said. And then there are also, of course, very important environmental factors. So early interaction at home. And we tried to control for this as well by quantifying parental education. Okay? And all these additional variables could not explain these positive associations here. And there were positive associations both with arithmetic and also with respect to visual spatial quantity processing. The arithmetic effect only trends towards significance. And I would assume, so my prediction is, as new data come in this summer, I would say that this effect will be significant as well. But we will see. Right now, it's not significant. But there was a strong positive association with visual spatial quantity processing. And it turned out that individual differences in the development of the right inferior parietal cortex explain almost a fourth of the variance in visual spatial quantity processing. Okay, and that's essentially my story for today, and this is also the take-home message I want to provide you with. So what have we found here? We have found a link between the gene LOBO1, which plays a major role for brain development. It's a classical neuronal migration gene, and brain network development of the inferior parietal cortex before children underwent systematic instruction in school. And based on these data, so based on these individual differences in gray matter volume in the inferior parietal cortex, we were then able to longitudinally predict individual differences in the visual spatial quantity tests. And yeah, that's essentially it. I would like to thank the Max Planck Society for funding this work, you for your attention, and I'm happy to try answering your questions. Thank you.
Very much. Great. Do we have any questions?
Hi, thank you, what do you think, what would you say are the implications of this for education?
So there is already quite some exciting work about the possibility of running early visual spatial quantity interventions. So there's for example this work by Karin Kuchian and colleagues from Zurich, who quite successfully applied number-line training to children that really targets these visual spatial quantity processing skills. And I think the message I want to send out here today is that despite the fact that there are some genetic constraints to some early genetic constraints to brain development, this is not set in stone, right? So there are other genes that also play a role for different aspects of plasticity. And I think it's really always worth and very important to apply these interventions, especially in populations that are already familiar or that have already risk backgrounds or difficulties in families. And so in other words, I think the main application is that we should take more care in making children school ready. I think we had this discussion already in the morning. And of course, we can do genotyping in all children to detect if they carry these genes. I think an important thing is to make parents aware of the fact that their own school history and their own performance is already a good proxy for this, right? So that would be one option. So the parental intervention tools. For example, this tool by Karin Kuchian that could be applied. It would be lovely if we would have, I think we had this discussion as well, if we would have randomized controlled trials really doing this. And then the second option, of course, would be to implement this in kindergarten settings, for example.
Thank you. There's a couple of questions in the middle. Emily?
All right, thank you, Michael, for this nice presentation. I have more theoretical question. Robo-1 has also been associated with dyslexia. And I wonder if you could elaborate a bit on whether your findings speak to some specificity or whether they are reflective of some larger association with academic learning, because we know that reading and arithmetic are highly correlated, the disorder score,
Yeah, that's a really great question. I think, so to be honest, I think what these genes do is really not specific. So it's really not confined to a certain cognitive skill or to a certain performance. So I think what these genes do is something very general. I think the only specific aspect that might be involved there is that these neuronal migration genes, so let me make the story a bit longer. These neuronal migration genes, so there are hundreds of them essentially. And what I think is what they are doing is they basically divide the brain into parcels. So each set of genes is responsible for neuronal migration, in different parts of the brain. But that's a very general anatomical mechanism. And now the link to reading and writing, in my view, is that Robo1 has a pretty confined effect on systems that are located in the inferior parietal cortex, or even more ventrally in temporal parietal areas, and that's where these difficulties where they might converge.
And did you have a question also? Yeah.
I think my question is much simpler. Could you go back to the slide where it showed the associations and explain the y-axis here?
The y-axis quantifies the gray matter volume, right? And so it's cut off. So as I said, that's a continuous value, right? So let me illustrate that.
So, but I don't understand where the gene and its possible mutations or lack of expression or expression
right.
levels
I
fits
see
into these graphs.
No,
That's where I'm confused.
That's a separate analysis now. So there were two steps in the analysis. The first step was to associate this genetic variance with brain variance, and this way we were able to identify this cluster here, right? So basically we searched the brain for a systematic link between DNA variants and gray matter volume variants to identify an area that is, whose maturation to some degree is under genetic control. And then we took this information into a separate analysis, what you can see here, and now did the second step, and this was to link these individual differences in brain development that are under genetic control to some degree to behavior. So that's a separate analysis. So in other words, the genetic information is kind of implicitly in here. Does that make sense to you?
Okay, so
use that logic?
All right, I got your logic, but what worries me is the huge standard deviations here. And if we're going to think about this kind of association between structure and some measure of intellectual function or whatever the task is, with large standard deviations, how do we tell a specific set of parents or a teacher that this has some specificity for that particular child's learning difficulties? Because the range here is essentially more than half of your overall scale.
Yeah. Yeah, I think that's an important thing, and I think a general issue here. So what can we do? I think we can do retest reliability analysis to show that there is some internal consistency in these measures. And we can do, we should go for more longitudinal analysis where you basically have like, an implicit baseline, and you can basically control changes that are never important. But yeah, standard deviation brain measures. I mean, there are efforts underway to improve these measures. So we could go for higher resolutions. So there are methods to improve signal-to-noise ratio. I mean, there are several factors influencing this. But in the end, you are right, so I wouldn't dare to claim here today that I can make specific claims here with these data regarding an individual child. I think one has to be really cautious here. So we are not there yet. We can make some group inferences, but I think it's kind of hard to make inferences about the individual. I think that's a general limitation we have to live with at the moment.
And actually some of these things are the things we might reflect when thinking about the sector as a whole, as we will be encouraged to do at the end of tomorrow and Wednesday. I think there was a question, but I'm trying to do the questions in the order they appeared. Do you stop? There was a question over there at the start. Has that been resolved? Okay, and then you had a question.
Hi, again, first of all, I quite enjoyed the fact that you explained running genes. I wonder if you could just help me join up dots slightly about kind of effect sizes. So you're saying you see a bigger effect size at the level of the brain, but you see very small effect sizes going from the genes to the final behaviour. Can you join those dots? Because I mean, that effect size is much larger than what we've seen in a genetics study. And presumably you don't see the link between, you've got genetic information on these children who've done behavioural tasks, and presumably you don't see much there, which is what we expect from the genetic literature. So how do you kind of square that circle in your head out? You just see much larger effects at the level of the brain. You're saying it's genetically informed?
Not sure if I understand your question correctly.
So, how you go from very small effect sizes at the level of from the genes linking to defining behaviour, but then you seem to be seeing larger effects when just going from genes to
brain,
brain, but much larger.
Yeah.
Have you got a read? I'm not interested in your
Okay, so what's the reason for that?
Yeah.
I think this relates to what I've said in the beginning. That's that it's simply there are a lot of studies that are running these tests between genetic variants and behavioral test scores, and I think the smaller effect sizes can be simply explained by the fact that this is somehow not a biologically grounded perspective on how genes work, because there's a lot of unexplained variants in between. And there's particularly neurobiological information, that kind of absorbs this variance. And I think it's, for regions of biological validity that you have these much larger effects on brain measures, because this is what these genes do, right? So they don't directly act on what we do, right? But on our brains.
Yeah.
And I think we've got time just for one more question. I think you have a question.
Hi, thanks for that, Miss. I was wondering about your thoughts on the shape of these associations. I think there are inverted U shapes.
Yeah, they reach a plateau at some point, the associations,
They reach a plateau.
Just because there's an upper boundary of the psychometric test instrument.
Oh, okay.
So there's-
Yes, that's instruments.
Yeah.
Thank you.